Now we move to the alignment step in this process of genome alignment sequence RNA-seq alignment. We talked about indexing and how to build an index to speed up the process, given that we have large genome files and align these reads to the genome, reference genome. So it makes sense, instead of reading the full book in terms of analogy from first page to page, from the first word to the last word, every time I want to search a term, well, let's break it down and look into the indices and jump to the right page. And now we can find, we are using these indices. There are strategies into how these indices can be used, defining the different types of algorithms. So when it comes to alignment, we can think of a series or steps that define the overall strategy for our alignment. Alignment is certainly the gateway to most bioinformatics analysis. In other words, I wouldn't imagine a bioinformatician, or let's say a skilled bioinformatician or an expert bioinformatician, without any knowledge in alignment. At least knowing the pipeline, and that things start with, after having some samples, we get the sequence read, we do some quality check, we may do some trimming to improve the data, and then here we apply some mapping to the genome. Eventually, once we do the mapping, we can count, right? Once you map to the genome, you define the regions. You sort of know this is the gene here. So let's count the number of reads that match here, come up with estimated numbers, and then use these numbers for statistical analysis. Here, we're talking about the downstream analysis. What we're focusing on here is the splice-aware mapping to genome, splice-aware mapping to genome. So I'm going to shift to notes here, although there are some slides for you that you can go through, but yeah, I prefer to go with this part for this part with some slides. So splice-aware mapping, splice-aware mapping. Okay, so this is the keyword here. Splice, it refers to the alignment of RNA sequencing or RNA-seq data to reference genome. Okay, while taking into account that there might be some splicing events, some events of splicing. Okay, let me explain this. What do we mean by this? With genes, as you know, are transcribed to mRNA, they go some splicing, where intronic sequences are removed. Okay. Okay, so pre-mRNA stage, and once we're going to get the mRNA, there is some processing that happens. So here's my question to you: when does that processing happens? We know that, and this is a treasure hunt question. We studied the DNA process transcription, right? Tell me exactly when this process happens, meaning this this process of splicing, okay, at what stage? And the second question, the additional question related to this part, how this happens, okay? How this happens, or what kind of enzymes are involved in this process. Okay, let me read your answers and thoughts based on the previous material or something if you want to look on and share your thoughts with me. But anyway, there are exonic sequences, and this is now, it's natural. Sometimes we kind of jump into key terms that may answer the treasure hunt question. But again, it's a way to participate and share your thoughts. So no worries. And for many of the questions, and kind of instantly you get the answer, which is still fine. Okay. So, and the remaining exonic sequences are joined together. So there are some sequences that are removed. Okay. If we get a splice-aware aligner and is not going to look into exonic or intronic sequences, then it will have an issue in mapping some reads. Let me illustrate this whole thing with some example. Okay, so imagine now it's just a hypothetical example. Okay, so imagine with me that we have some gene sequence. Okay, and this some gene sequence is made of, let me write some alphabets here, A, T, G, C, G, okay, G, C, G, let me put T, A, and next, I'm gonna do this. I'm gonna use a different color, and then I'm gonna go back. So here we go. We have some intron here, some intron, okay, some intron here. And then what I'm gonna do is I'm going to continue GACG, GACG, and AT, and we keep going with the gene. So this is the gene. Now, we know after splicing, the intronic region is going to go. So this is the gene, which is part of the DNA. So now let's move to the second stage, which is the mRNA. Okay, remember transcription. Okay, now after splicing, the intronic sequence is removed. So what do we end up with? Well, this is what we end up with. Okay, we're going to write down everything consecutive to each other. So this is AT, G, C, G, T, A. This is from here. And then G, A, C, G, A, T. So we remove this part and then we join these two together. Okay, so this is the mRNA now. Nice. Now what do we have? Well, we're going to have some reads. Okay, we're going to have some read. Imagine now the read we have is made of the following. Some sequence, and in this sequence, we have G, C, GTA, oops, GC, GTA, GC, GTA, GAC, okay? And so this is our read now. Cool. So this read actually spans the spliced junction, this area. So let me highlight this, make it clear for you. So now what do you see here? Well, there's GCGTA. So there's this part here, okay? This is where we have the read from, right? Okay. And then there's GAC. GAC. So remember, we take these reads and we want to map them to the genome sequence file. So if the algorithm that's going to work on this is going to directly only match exactly the same sequence, it can start by that. So you can define a strategy where you say, okay, let's try first to see if there is an exact match, and then we are done. Nothing wrong with that. But what if you don't find? Okay, are you going to throw it away or consider that an error? What are we going to do? It could be an intro, because when we map to the genome reference file, we have introns there. And the reads are a reflection of what we receive from the mRNAs. Okay, so in this case, how can we do this mapping? This is the challenge here, as we have a gap. So are we going to say, well, push this away a little bit and push these from each other and then consider that a mapping, or what are we going to do? So these are the kind of questions that one need to handle when we work with what's called alignment. Otherwise, an unsplice aware aligner that just discard this whole issue of splicing and if introns, may misalign, may fail to align, may break to read the read into fragments, okay, and would align them separately. So anyway, now there are several popular tools that take into consideration this scenario of splice-aware mapping, including STAR, the one that we'll use for the in-class activity. There is STAR, there is Top Hat tool, there's HiSat. All of those are splice-aware, okay? Are splice-aware. Now, let me dive a little bit with these notes as well into STAR. So STAR or splice transcripts alignment to a reference. That is STAR, splice transcripts alignment to a reference, okay? It is a popular splice-aware aligner that efficiently map RNA-seq data to a genome. So it requires a genome file. And it's particularly well suited for detecting splice junctions, these areas of interest. Okay. So again, if I have some gene, where's some sequence, and then some intron, okay, this is my coming from my genome file, and then I have my read that I want to map, okay, and again I have some read here like the previous example, G C G T A G A C. Okay, so what STAR is going to do? STAR is going to go through a procedure of specific steps. What is the first step? This is my treasure hunt question for you. What do you think is going to be, what's going to be the first step? Tell me. Share with me your thoughts. Again, there's no wrong or right answer. Now, you may say, why I'm asking this question, I'm just explaining a new algorithm where if I'm asking this question, means we have discussed this somewhere. Okay? I'm going to give you a moment. The first thing is indexing the genome. Okay, when I make the process efficient. So before alignment, STAR creates an index of the reference genome. That's why when you go to the in-class activity and you read through it, you will find this written that the class instructors did already prepare the index for you, because this steps takes time. And we don't want everyone to try it since we're using the same reference file for the genome reference file for the exercise. So it is sufficient that only one would do it and then all can use the same index file. In other words, if you're in your lab, so one can be assigned this task of building the index and then everyone else can use this index from now on. Okay, so during the step of indexing the genome, STAR program, software, or algorithm also detects potential splice junctions. How it can do that? Okay, well, it is using genome annotations, or maybe it can predict these areas of splice junctions. So we're done with the first step, and what is going to be the next step? And again, this is another treasure hunt question for you. What do you think is going to be the next step? I'm going to give you a moment. Share with me one keyword, okay? It's fine. Well, this is seed mapping. Seed mapping. So STAR first looks for seeds or smaller fragments from the read that match the genome. Okay, so for example, for our read here, which is this one, STAR might identify seeds like GCGTA. GCGTA might consider this as a seed, one seed. So GCGTA, and so goes directly and starts mapping this. And GAC, this could be another seed, GAC, okay? So this is the second step. Now we're going to move to third step. If this was a splice-aware mapping or alignment tool, what do you think is going to be in step 3? And again, it's another treasure hunt question. I want you to kind of predict what's going to be this step. Okay. I'm going to give you a moment. Okay, it is splice junction search. Okay, well, here I'm gonna get your attention. If any of these seeds does not align continuously to the genome reference, star checks if the seed's alignment extends to a known one. Okay, or a predicted splice junction. So let me repeat this. It's either we have an option where the seed is going to align continuously with no issues, because it is good. So star is going to do this, check this. Or it does not align continuously. In this case, we need to check the seed's alignment if it extends to a known or predicted splice junction. In other words, we're trying to know if this sequence is related to some known documented beginning of introns, for example. If we previously know that GCGRA, for example, usually is followed by intron, so yeah, we know this. This is an area that we need to keep an eye on, okay? Because GCGTA maps directly before an intron, and maybe GAC maps directly after the same intron. So if we know this information, or we can predict this information, we can locate these junctions in our read. Once that is done, we are good to go with what? With read alignment. With read alignment. Okay. So star then now knows that our read likely spans a splice junction. So it's going to align this here to this area here. Okay. Although the initial read was like this one, but star was able to figure out, let's split this here, take this part to this one, and take this part, align it to this area. Okay, so the beauty of star is in drawing this line, knowing where to split these seeds and given read. Eventually, then we're able to map this whole read to the genome. Otherwise, we may fail doing this. Okay, now in reality, star has to handle numerous complexities, edge situations, exceptional cases, including multiple potential mapping locations for a read or sequencing errors and vast number of reads. And actually, star uses a graph-based approach to efficiently and accurately address these challenges. Okay, but in a nutshell, these are the general steps. Now, two final concepts I want to introduce here as to, close the discussion on star alignment as an example of an alignment software is MMPs, okay? MMPs, which is maximal mappable prefixes. So when star aligns RNA-seq to a reference genome, it attempts to maximize the alignment's length, okay? However, when a read spans a splice junction or has mismatches, continuous alignment might not be possible in that case, as mentioned earlier. Okay. So an MMP is the longest prefix. That is the portion of a read starting from the first base that can be mapped to the reference genome without any mismatches. So that is the maximal mappable prefix. So as an example, if a read spans a splice junction, the MMP would be the portion of the read that maps up to the splice site. Okay. So star kind of also try to find these areas. And again, as I said earlier, is here we're trying to draw. It's trying to draw this line. Another concept is what's called soft clipping. Okay. So this is a concept used in sequence alignment to handle portions of a read that do not align to the reference genome. Okay. So when a read is soft clipped, we have, let me write this down. Okay, yes, we have soft clipping and we can also refer to hard clipping. So anyway, when a read is soft clipped, certain bases at its beginning or end are clipped off or ignored during the alignment, but they remain present in the output through certain formatting, unlike hard clipping where they would be removed. Okay, think about trimming adapters or certain areas. So sometimes star may take a portion of a read that it cannot align to the genome, maybe due to sequencing error, some genuine genetic variations or other reasons. For some reason, that portion can be soft clipped, removed. Okay, in other words, and then again, if a read spans splice junction area, star will align the MMP to the genome, identify the splice junction, align the remainder of the read after the splice junction, and if there are bases in the read that cannot be aligned either due to mismatches or other reasons, they might be soft clipped in the final alignment. So anyway, I hope this summarized the few ideas that I wanted to cover. Okay, and finishing this part of the module. Let me see if we want to discuss something more. Yeah, one of the things that it's in principle what we are dealing with is also alternative splicing. Okay, alternative splicing. Now, we may face cases with alternative splicing, like when exons are there and introns are removed. Exons, for example, can flip in order towards generating the pre-mRNA. So if I have, for example, this is exon 1, then there's some intron here, and there's exon 2, some intron here, and exon 3. Now, part of the process is to remove these introns. But again, this exon may jump here, and so we have exon 2, and then followed by exon 1, and then exon 3. And we may have sometimes exon 3, then followed by exon 1, and then exon 2. So those are going to do some variations in terms of the sequence that we are looking at. So alternative splicing is a molecular process by which a single pre-mRNA, and again, I'm saying pre-mRNA molecule, can be spliced in different ways to produce multiple distinct mRNA molecules, multiple distinct mRNA sequences. These variant mRNAs can then be translated into different protein isoforms. Okay, so we have protein isoforms in this case. This allows for a wide diversity of proteins from a limited number of genes. So we end up with some diversity of proteins, but they are still isoforms. Okay. So in this case, this is one of the, there are many conditions or cases why we end up having alternative splicing. Okay. For example, exon skipping, or what is called cassette exon. In that case, let me write it down. Exon skipping. In that case, guys, an exon may be included in some mRNAs and skipped in others. And this is the most common type of alternative splicing. Kind of, well, you know, this could be also skipped. This is one of the common cases, exon skipping. There's intron retention, where an intron is retained in the final mRNA instead of being spliced out. Okay, intron could stay. Okay, and alternative 5' or 3' splice sites, and so forth. Mutually exclusive exon, only one of two possible exons is included in mRNA, and so forth. Okay, these kinds of exons arrangements are going to lead us to different sequences in terms of RNA. So this is one of the challenges. This alternative splicing is going to give us some ambiguity as we try to do the alignment. Okay, makes it a bit challenging. However, we introduce a software. We describe the star. Star will try its best. And again, usually what we do, as I said, we may keep an eye on these developments of these algorithms. Every now and then, there might be some update. Look into the quality. It is common sometimes to make a comparison between these different tools. And as a bioinformatician, go and given that you have specific experiment, why not to run the several pipelines and see the one that gives you the best results in terms of quality and downstream analysis? Yes, it could be a demanding process, but then it could set up, the workflow for the next project or so. That's it for this module. There is an in-class activity which is up and running now. I just couldn't yet get some access to it with resources. Just today, I mean. I was able to get access to the login node. I couldn't get an interactive session to show you some of the steps. I would highly advise you go and read through the in-class activity and follow the steps there. And given that we had previously the in-class activity, that is last week's section where I've introduced you to the terminal, I think now you can take over this part on your own. So best of luck, and I hope you communicate if you have any questions.